my advisee. And now that I've been on sabbatical twice, I'm losing her, so I'm like, if anybody needs an advisor, just let me know, because I'm kind of running low on advisees. That's just a joke. But anyhow, it's been a pleasure having Melissa in a variety of different classes. She's been terribly organized as an advisee, coming in knowing exactly what she wants, and then getting it. Comes and asks for it, and it's great. She's from Wausau, Wisconsin, so it's not too terribly far. And that she is going to be going to Chicago to become a lab medical scientist this coming fall. So without further ado, I will turn it over to Melissa.
All right, so the idea for today's talk arose from a walk through the grocery store of all places. And I was walking through the store and I saw a lot of products that said things like sugar-free or no calories, or even more interestingly, no aspartame added. And it got me thinking about discussions we had in Biochemistry 2 over the efficacy of artificial sweeteners, as well as talks I had seen on the news of the utilization of artificial sweeteners in general. So I did some digging, and to be honest, I found a lot more bad than good. Artificial sweeteners were associated with the very thing they were intended to prevent, obesity. And this could be explained in some part by our neglect of an important part of the body, the gut microbiota. In the last century or so, we have seen profound changes in the way that we eat, we live, we work, the way we have been treated medically. And with these changes, we have seen an upward rise in non-communicable postmodern diseases, including things like allergies, asthma, obesity, and type 2 diabetes. Specifically, these changes in diet were related to changes in diet, like our Western diet of high processed foods, high fat foods, low plant materials, as well as changes in an increasingly sedentary lifestyle. In fact, obesity then is one of the greatest threats we face today. More than 1/3 of adults in the United States are obese, and it's projected that by 2030, 2.16 billion adults will be obese. But what we're looking at isn't just a problem of obesity, but a greater health problem at large. Obesity in some ways can be considered like the gateway disease of non-communicable diseases, leading to problems with your heart, your lungs, your heart, hypertension, stroke, cataracts, liver disease, arthritis, cancer, and one of the biggest problems we're going to see associated with obesity is type 2 diabetes. And as many of you may know, type 2 diabetes involves resistance to insulin, so the body is unable to uptake glucose. So let's look at this a little bit more specifically. In type 2 diabetes, you're going to have a meal, and your stomach's going to convert that meal into glucose. Glucose will enter the bloodstream, and we have the pancreas producing insulin. However, in type 2 diabetes, insulin resistance means that the receptors meant to bind to insulin either are not expressed on the surface of cells or do not bind insulin normally. As a result, there's no signal for glucose to be transported into the blood, out of the blood effectively, and as a result, we have an increase in levels of glucose in the blood. This ultimately is going to lead to glucose intolerance. And what I mean by that is heightened levels of blood glucose concentration over a longer period of time. And so for a normal individual, if we have a meal, we're going to have a spike in glucose, but our insulin is going to actually allow that glucose to be transported out of the blood. And so we have a more healthy level of glucose in the blood over a longer period of time. In a diabetic, however, this level is significantly raised, and it's going to be even more so with the ingestion of glucose, and this is going to take a much longer time for it to get to a steady state for a normal type 2 diabetic in this case. So to combat the problems of type 2 diabetes, obesity, and glucose intolerance, many individuals have turned to artificial sweeteners as their solution. And so what I mean by that is non-nutritive or non-caloric artificial sweeteners. So these sweeteners are going to contain little to no calories. And specifically, we have 5 who have been approved by the Food and Drug Administration: saccharin, aspartame, acesulfame, potassium, sucralose, and neotame. And the idea behind these non-nutritive sweeteners is that if we replace them in our diet in the place of sugar, we're going to reduce caloric intake and thereby reduce weight gain. But as it turns out, the problem isn't so simple. This is taken from an article by Yang talking about the neurobiology of artificial sweeteners, and he sees that in the last 50 years or so, we have seen a steady increase in the percent of population consuming artificial sweeteners from about 3.3 to about 15.1%. In red, we can see how the increase in obesity, so a body mass index of greater than 30, has increased steadily during this time as well. In purple, we're looking at new products containing artificial sweeteners, and this has significantly increased in the last 10 years. In fact, with this steady increase in the last 15 years, we also have seen a steady increase in the percent of population consuming artificial sweeteners, and more alarmingly, the percent of population who has become obese. And so this suggests perhaps we have an association between artificial sweeteners and obesity, and perhaps that artificial sweeteners might play a causal role. And many individuals have many hypotheses for this association, one of which is that non-nutritive sweeteners are already used as a dietary aid. So in this case, we're not looking at artificial sweeteners as playing a causal role, but what we're seeing is a reverse causal role. So if I'm overweight and I'm looking to reduce my caloric intake, I might turn to artificial sweeteners, but since I'm already gaining weight, it's going to be associated with this weight gain anyway. However, artificial sweeteners have been associated with weight gain in a dose-dependent manner, suggesting that there's more going on than just this reverse causal role. So something else that might be going on is that we're uncoupling sweet taste from calories, leading to metabolic derangements in the body. And what I mean by that is we have sweet taste receptors in our mouth who are going to, in essence, taste the sweet taste coming in and lead to a variety of signal transduction reactions that plan for the presence of energy in the body. And when no energy comes, we're going to have a lack of homeostasis in the body. We're going to disrupt that homeostasis, and we're also going to have problems with desensitizing the system. We also have evidence for sweet taste receptors in the gut, suggesting that this is another pathway or place by which we can't actually escape the presence of these sweet tastings in the body. In fact, this image here that I made would be a very simple example of a sweet taste receptor in the gut epithelium. The purple would be a sweet taste, and this is going to initiate the release of hormones eventually that are going to regulate insulin release. This is ultimately going to mean we have rising insulin levels even though we have no glucose in the body. So we're again altering that energy homeostasis that is meant to help our body rather than hurt it. So the last hypothesis for artificial sweetener association with obesity is that they actually are going to alter the gut microbiota, leading to obesity. And this is the one we're going to be focusing on today. But some of you might wonder, what exactly is the gut microbiota? Well, the gut microbiota consists of 10 to 100 trillion microorganisms that reside in our gut. And if you think about it, that means that these microorganisms substantially outnumber the human cells in our body by a lot. And as a result, we would begin to think that maybe they play a significant role in our body. And it turns out they do. We have co-evolved with these organisms so that they're getting the necessary energy sources they need from our body, and we are getting aid in the digestion of materials we would otherwise be unable to digest. So, for instance, like plant saccharides that we would not be able to digest. In this way, we might consider the gut microbiota as an important metabolic organ in the body, something that plays an important role in the metabolic processes that we can't neglect when we start to consider the incidence of disease, specifically in this case obesity, as well as the treatment of that disease. So in order to understand how the alteration of the gut might lead to obesity, I think it's important to characterize the normal gut microbiota. So the normal gut microbiota consists of greater than 1,000 microbial species and greater than 50 microbial phyla. And of these microbial phyla, we have the main composition is going to be Bacteroidetes and Firmicutes. So if I were to take a random sample of microbes from the gut, chances are it's one of these two. However, despite this general composition, we're going to have significant variation amongst individuals according to age. So infants are going to have significantly less diversity than an adult, for example. Genetics, there's an idea that the mother is actually able to pass down some of her gut microbes to her child through childbirth. Environment, if you are in a different environment, we might expect different pathogens to play an important role in the composition of your gut as well. Diet, if we're putting different things in our body, we would expect to be altering the very things that play a role in the digestion of these items. So if we give our microbe a cheeseburger, we're going to get different things in our gut microbiota than something else. So ultimately, what does this mean about alteration of the gut microbiota and obesity? Well, if we think about it before, I said that the gut microbiota plays an important role in degrading things we would otherwise be unable to digest, and as a result, we can increase energy intake. So the gut microbiota itself can be considered an environmental factor that can regulate fat storage and lead to the obese phenotype, especially when it contains the bacteria that are going to play an important role in these processes. So in order to understand this, I think we need to look at the obese phenotype as it looks like in the gut microbiota. Specifically, the obese phenotype in many individuals in the United States is going to arise from the Western diet. And again, I said that was going to be high fat, high processed foods, low plant material, and very little variation. And with this lack of variation, we're going to actually see a decrease in microbial species, decrease in microbes in general, as well as a decrease in Bacteroidetes and an increase in Firmicutes. What this means is we're going to be altering the very composition of those two main phyla that we saw in the gut earlier, suggesting that we're going to be changing the byproducts of that gut. One thing that implicates the role of the gut microbiota in the incidence of obesity is that we can actually classify individuals as obese or lean solely on the basis of their gut with 90% accuracy. Beyond this, another thing that implicates the role of the gut in the presence of obesity is that we can actually transplant the obese phenotype into a healthy, lean, or germ-free individual and induce that obese phenotype in that individual. This is huge. What that means is that without altering the environment or the diet of those healthy or germ-free mice, we can actually induce obesity simply by transferring the gut microbiota through a fecal transplant into those mice. So how exactly does this happen though? It's going to have to be due to some of those byproducts of those bacteria in the gut. And one of these is going to be short-chain fatty acids. And so an example of short-chain fatty acid would be acetate, or as many of you may know it, vinegar. So short-chain fatty acids have actually been found to be in heightened levels in obese individuals. And so it's been suggested that this transfer of the obese phenotype into these healthy, lean, or germ-free individuals has been mediated through this increased absorption of monosaccharides and short-chain fatty acids. But what exactly is their role in obesity? How does this happen? Well, it has to do with short-chain fatty acids as a great energy source. It turns out that short-chain fatty acids are used by the liver to undergo gluconeogenesis and lipogenesis. So in gluconeogenesis, we're going to be creating glucose de novo, and in lipogenesis, we're going to be starting to create more and more triglycerides. So we're going to have short-chain fatty acids being made into triglycerides, and over time, as these triglyceride levels accumulate, lipases are going to come in, specifically in our case, lipoprotein lipase, and break down these into fatty acids, which then are uptaken by cells in the body and increasing adiposity. Let's look at this a little bit more specifically according to what's going on here. It turns out that increased triglyceride production is associated with decreased expression of a protein called fasting-induced adipocyte factor, or FIAF. And fasting-induced adipocyte factor is actually an inhibitor of lipoprotein lipase. If we're going to decrease the expression of that protein FIAF, we're going to increase the activity of lipoprotein lipase. Now, lipoprotein lipase mediates the cellular uptake of those fatty acids and increases adipocyte formation, so ultimately giving us weight gain. Specifically, we can associate FIAF as directly playing a role in the incidence of microbial adiposity by looking at germ-free FIAF deficient mice, so mice who do not have the genotype for that protein FIAF, and discovering that they actually have just as much fat mass and body weight as colonized mice, so mice who do have a gut microbiota. This suggests that it is directly through the decreased expression of this FIAF that weight gain is mediated. Beckett and his colleagues in an experiment in 2014 actually looked at this very idea. Specifically in this image, we're looking at epidemial fat pads of 8-week-old mice. On the left, we have germ-free mice. On the right, we have conventionally raised mice, so mice who are given a normal diet. And in the center, we have conventionalized mice. So these mice have had the gut microbiota from conventionally raised mice transplanted into their own guts. As you can see, both the gut microbiota containing mice have increased weight gain, suggesting that it is directly through the microbiota that this weight gain occurs, especially since we transferred the gut microbiota of the conventionally raised mouse into the conventionalized mouse and saw the same effect. Now, looking at FIAF, they actually looked at LPL activity. So again, you would expect LPL activity to increase with FIAF suppression. And so specifically, they looked at FIAF activity on the y-axis, and in the white we have germ-free mice, in the black we have conventionalized mice. So as you can see, in the epidemial fat pads, LPL activity increased significantly, as well as in the heart, suggesting that FIAF is directly mediating, or the suppression of FIAF is directly mediating this increase in weight gain over here due to the increase in LPL activity. They also looked at the genotypes of FIAF mice, and as you can see, they compared these genotypes in germ-free mice, compared it to increase in body fat of conventionalized mice, so those mice that did harbor a gut microbiota. So conventionalized mice were, a conventionalized mouse had very little increase in body fat compared to a mouse of a FIAF deficient genotype, suggesting that it was through this suppression of FIAF that microbial induced obesity was occurring. And so we began to see FIAF as an important mediator or regulator of weight gain in the gut microbiota. So let's look at this a little bit more in general. What we're looking at is the Western diet is going to alter the composition of the gut microbiota, leading to increased production of short-chain fatty acids. This increased production of short-chain fatty acids is going to actually lead to increased production of triglycerides, which, as we know, decreases expression of fasting-induced adipocyte factor. Fasting-induced adipocyte factor is an inhibitor of lipoprotein lipase, so that's going to increase activity of lipoprotein lipase. And as we know, lipoprotein lipase increases absorption of these fatty acids and increases adiposity, ultimately leading to weight gain. With increased weight gain, we're going to have risk of obesity, which again is a risk of type 2 diabetes. In type 2 diabetes, we have insulin resistance, which means we have decreased ability to transport glucose into the body, and as a result, increased level of glucose in the bloodstream, and ultimately glucose intolerance. So taking these ideas and putting them together, Jonathan Suez and his colleagues wrote an article on how artificial sweeteners might induce these changes in mice, specifically through the gut microbiota. And this was published in Nature in October 2014. So looking at this article, as well as considering what we've already learned, I put together a hypothesis for how artificial sweeteners might induce obesity in mice. So, as the gut microbiota already acts in a way that induces obesity, especially when perturbed, Suez and colleagues proposed that non-caloric artificial sweeteners similarly modulate and disturb the gut microbiota, leading to increased susceptibility to metabolic disease, thereby initiating a series of events that are contradictory to the initial purpose of non-caloric artificial sweetener utilization. To put it more generally, artificial sweeteners are going to cause an altered gut microbiota leading to obesity. So I thought that Suez and his colleagues did a variety of great experiments to test this hypothesis, and they were illustrated really well in an article reviewing their experiment by Bakovich and Glaser. So in one of their experiments, they gave mice artificial sweeteners in their drinking water, and control mice were given either just water, water and sucrose, or water and glucose. This treatment with artificial sweeteners eventually led to an altered gut microbiota, which led to increased in glucose intolerance. To determine whether or not this was specifically mediated by the altered gut microbiota, they added antibiotics to these artificial sweetener consuming mice and found that glucose intolerance was actually reverted back to its normal level. Again, to determine whether or not it was the altered gut microbiota leading to this glucose intolerance, they also transferred this gut microbiota into germ-free mice and found that glucose intolerance was inducible in these mice due to the altered gut microbiota. So let's look at their results a little bit more specifically. In this case, what we're looking at here on the y-axis is blood glucose concentration over a time period of 120 minutes or 2 hours. And again, we saw that in glucose intolerance, we would expect heightened levels of blood glucose over a longer period of time. As you can see in blue, what we're looking at is artificial sweetener consuming mice. And as we see is that the blood glucose test shows that we have an increase in levels of blood glucose over a longer period of time in these mice, suggesting the incidence of glucose intolerance in these mice. Especially if we compare it to the black, which is our control mice here. They also added antibiotics, seen in orange and gray, and this level of glucose intolerance was reverted back to normal, suggesting that it was directly through this change in the gut microbiota that this glucose intolerance occurred. One thing to note is that saccharin, the blue circles, had the highest effect of all of these artificial sweeteners, and as a result, they chose to use it for the transfer into germ-free mice. And they compared this against glucose-consuming mice. And again, we're looking at blood glucose level over 120-minute time period. And what we see is, yes, these artificial sweetener consuming mice have a gut microbiota that is transferable into germ-free mice that leads to the same level of glucose intolerance. Again, confirming the role of the gut microbiota in this change. What exactly can we say about obesity? Well, remember what we were talking about before of how short-chain fatty acids have an important role in this process? Well, these short-chain fatty acids are going to be created by glycan degradation enzymes who are going to degrade polysaccharides and monosaccharides that are coming into the gut. Specifically, we're going to have bacteria called glycan degrading bacteria who are going to possess these enzymes and use it for these processes. Suez and his colleagues were actually able to look at the gut microbiota composition with these different enzymes at week 11 compared to week 0, so before any treatment occurred in these mice. And here we're looking at saccharin-consuming mice, glucose-consuming mice, and only water-consuming mice. And then on the right here, what we see is a legend for the change in expression. And as you can see, red is going to mean the biggest change in expression in these enzymes, specifically. As you can see in saccharin, this is the row for saccharin, we see the greatest levels of red, suggesting the greatest amount of changes. This isn't surprising. We've already seen that saccharin is going to induce changes in the gut microbiota. However, what was interesting is that all these red boxes are going to be associated with enzymes specifically that are going to lead to be associated with glycan degradation, suggesting the presence of bacteria that are associated with glycan degradation, specifically making us wonder if short-chain fatty acid production has increased. And it turns out Suez and his colleagues also wondered this, and they actually tested this in mice at 11 weeks, compared glucose in black to blue saccharin-consuming mice, and the concentration of acetate in the feces as well as the concentration of propionate. As you can see, the levels of short-chain fatty acids in both these types have increased in saccharin-consuming mice compared to that of glucose-consuming mice, confirming that artificial sweeteners are modulating the gut in a way that we have changes in bacteria that are capable of leading to these increased levels of short-chain fatty acids that are ultimately going to lead to weight gain. Suez and his colleagues also were able to look at human subjects, not just mice, which is pretty cool. They looked at specifically 7 healthy human subjects who do not normally consume artificial sweeteners on a normal basis or artificial sweetener-containing products. And they gave them saccharin for 7 days, according to the FDA's regulation for maximal intake, I believe. And what they found was that most individuals responded just like the mice had. They had an altered gut microbiota that was transferable into mice, inducing glucose intolerance as well as inducing glucose intolerance initially. However, 3 out of the 7 individuals did not respond to artificial sweetener treatment during this time, and they possessed a normal gut microbiota that, again, when transferred into germ-free mice, had a relatively normal level of glucose tolerance. This suggests what we have going on is something in the gut microbiota initially that makes them more susceptible or resilient against the influence of artificial sweeteners. So let's look at the gut microbiota of these individuals. Here, what we're looking at in the different colors are associated with different microbial orders of the gut. So different colors are going to mean different composition. On the right, we have responders. On the left, we have non-responders. So if we look at responders from day 1 to day 7, we see a significant change in the composition of that gut. Again, this means, okay, so the gut's being altered, and this is the way by which we have. This is the way by which glucose intolerance occurs by the alteration by these artificial sweeteners. However, if we look at non-responders have very little change in their gut from day 1 to day 7, suggesting that we are seeing, suggesting that these gut microbes are perhaps more resilient against artificial sweeteners. In fact, if we look at day 1 of non-responders compared to day 1 of responders, the gut microbes in this case are significantly different, suggesting that maybe non-responders have more K species, more competitive species, capable of defending themselves against the influence of artificial sweeteners, or something in responders makes them more susceptible to artificial sweetener influence. I don't know. But that is something that would require perhaps more research. One thing I would like to mention is that this is only a 7-day study. Perhaps we would see greater impact in non-responders if we had looked for a longer period of time. Also, we're only looking at 7 human subjects. Perhaps if we had looked at more, that would have been an interesting thing to see. I don't know how ethical that would be, considering that we are inducing glucose intolerance in these individuals, so I don't know if that's something that we would want to do, but it's something to consider when we look at these results. Again, as I had said before, we actually transferred the gut microbiota of these responders versus non-responders into germ-free mice, specifically at day 1 and day 7. And as you can see in responders, the blood glucose concentration is significantly greater over a longer period of time compared to that of day 1. However, in non-responders, there's very little change. One thing, again, to note is that at day 1 of responders, we have significantly greater glucose concentration over a longer period of time compared to that of day 1 of responders. Again, suggesting that there's something different going on here between the guts of these individuals. However, ultimately, most individuals did respond to the influence of artificial sweeteners, and we know that they certainly have the ability to change the gut microbiota. What this means is that fake sugars aren't doing so well. They're altering the composition of our gut, and through that alteration of the gut, they're leading to an increased risk of obesity and glucose intolerance. That's bad news. So that makes me wonder about artificial sweeteners in general. Clearly, I don't believe that these are a good idea. Is there a possibility to make artificial sweeteners that are going to not modulate the gut? I don't know, especially since these products are going to be coming through our body and making direct contact with the gut microbiota. It seems like a difficult thing to do. And also, looking at these responders versus non-responders helps us consider how personalized nutrition is going to lead to personalized medical outcomes. We have a lot of different things going on in our body, and one of these is going to be a different gut microbiota, which is going to make us more or less susceptible to the incidence of disease, and as well as, in our case, the impact of artificial sweeteners. This also, by that same token, means that we might use this to our advantage in creating personalized medical plans or personalized treatments according to the composition of one's gut. In this way, we might be able to better target treatment for you because of your diet than for me and my diet. Some of these things are important to consider when we consider obesity, but also other diseases as well. Just because I have linked artificial sweeteners to the incidence of obesity through the gut microbiota doesn't mean I believe this is the sole pathway by which obesity occurs. It might be through a variety of reasons, specifically those sweet taste receptors in our mouth and our gut. There might be evidence, again, for the use of artificial sweeteners as a dietary aid. But there might be other reasons, like mTOR autophagy, that we haven't really considered at all. But for me, I think that artificial sweeteners are bad news. They're not good. They're dangerous, they're unhealthy, and they're a whole lot of trouble. And to be honest, that's not a risk I'm willing to take.
Not everybody at once.
I guess my understanding of some of these artificial sweeteners is that they make them have 0 calories by taking sugar or some sort of sugar like molecule and then substituting individual atoms onto it. So like taking like the H off of the sugar and then putting like a chloride or something onto it. Do you think that maybe that doing that is making is what's changing the microbes, since they're obviously developed and they've co-evolved with us to do certain things, and so by changing these sweeteners, it's really messing with them because we're like forcing them to change.
Yeah, I think that does play a huge role. I don't know for sure because I didn't specifically look at how they modulate the gut microbiota. We're looking at that they do modulate the gut microbiota, but I think that's a really good idea for it. Yeah?
I don't know if you can answer this, but I'm hoping you can. What about like low calorie drinks? Do those also microbiota?
I would think so, and especially since what I was talking about with those non-caloric artificial sweeteners, some of them are low calorie, like the ones I think some of the ones I explained earlier do have low calorie.
So the cases they did with the mice and the humans, I might have missed this. Did they study what would happen, especially with humans, after they were taking off this change of diet? Is there like a reversion back to the original microbiota, or?
No, they actually didn't do that. No, I didn't see anything about that. They looked at what their guts looked like before, but not after.
In the same way, you talked about how the treatment of antibiotics could bring that back. I was just kind of curious about timescales, so I just want to make sure it's clear in my mind. So was it 11 days that they treated with the glucose and then they tested it? Or timing of how feeding mice glucose or the NASs led to this new microbiota? How long was that time frame?
Oh,
and then
well,
after that, how long did they treat with antibiotics to get it return back to that normal what you showed?
okay. So I think what we're looking at for the first experiment you're talking about, they actually looked at what the evidence from that is from 6 weeks of treatment, I believe. So these mice have actually been consuming artificial sweeteners for a really long period of time. For the antibiotics, I can't say for sure when. I don't remember. I'm sorry when
Yeah,
that was.
I'm just curious about how responsive it was.
Yeah.
And that would be very interesting to me.
I can't remember.
I have another one.
Okay.
You had said that there was some indication that this was that it was genetics because of that when a mother could pass it during childbirth.
Mm-hmm.
I didn't quite understand that statement.
There's evidence that in vaginal childbirth we can actually transplant gut microbes into the child.
Maybe I misunderstand genetics. That would indicate that like HIV is not genetic, but you can pass it on.
Right. I guess that's the way that when they discuss genetic, that's the way that they have been describing it in all the papers I've been reading, is that the direct relation to your mother and passing down from child to mother to child through that way.
Thank you very much. Tuesday, 11:10 right here. Make sure you sign up for the picnic outside the Stockton. Actually, yeah.